that we can produce for pharmaceutical markets. And so for cosmetic and pharmaceutic, for example, we develop active ingredients, we develop probiotics or metabolic derivative. We work on biotransformation, meaning we use microorganisms for the fermentation of plants to produce specific ingredients. We have a recent project on skin microbiota. And also, we have recent project on the production of live biotherapeutic product, meaning it's a pharmaceutic using a live microorganism. Another market for us is agronomy market, for which we develop biofertilizer and biocontrol products. Here we already have formulated product composed of bacteria and fungi, and already registered in France and in Europe, in some countries in Europe. Those products are very interesting for the management of the fertilization, to improve soil microflora, but also we have a project and registration ongoing on biocontrol of plant diseases. And also we have developed a formulation for decomposition of organic matter. As I said just before, we have a company, Green Sea, dedicated to algae, and we are developing a portfolio of products composed of original algae. So I will do a focus on biosimulation and biocontrol. Our microorganism are alive microorganisms, so we market product with shelf life, interesting shelf life, and the microorganism alive that will have an impact on the crop residue, on the organic matter of the soil, that will improve nutrient assimilation. Thus, we will have a better resistance of plant to abiotic stresses, and we will have the capacity to improve yield and crop quality. And in fact, globally, we will work on the recolonization and rebalancing of microbial life in the soil. So our biosimulation products are today dedicated to the soil and to improve the life in the soil. Also, I have to indicate that application of microorganisms in the soil is very important because we have a physical barrier, like a biofilm, which will be a formation of a protective layer around the roots and traps of pathogens. We have mechanism of spatial competition or food competition. So with our product, we will add beneficial microorganisms. We will limit pathogen invasion. And also we will improve the production of metabolites that will have inhibitor effect on pathogens. For example, we have a product with the Pseudomonas fluorescens that can produce phenazine, and which is efficient to control some food rot. So we work on several microorganisms in the lab, but to date, we have formulated product composed of bacteria, Pseudomonas fluorescens, Bacillus methylotrophicus, Azotobacter vinelandi, and also we market fungi. It's a Trichoderma asianum and Phanerochete chrysosporium. If I take the example of Pseudomonas fluorescens, this strain, the strain we produce will be interesting for stimulation of seed germination and the seedling emergence. Why? Because the bacteria is able to produce IAR, meaning phytoauxin. The microorganism is able also to capture iron, so the strain will help the plant to develop. The strain will be able to protect the root with a biofilm, and when thus we will improve the growth of leaves and roots. And globally, we observe resistance to abiotic stress. And also, depending on the microorganism, we can have biocontrol activity. The mechanism associated with biocontrol activity are antagonism with plant pathogen, or competition for space and food, or plant systemic resistance induction. So we have ongoing project to develop biocontrol product on wheat, on rapeseed, and on grapevine trunk diseases. And then in another market, in environment, here we develop cocktail of microorganism, combination of microorganism to treat industrial effluent for the treatment of polluting compound, also for the removal of bad odors or for composting processes. So in fact, Biovitis would like to share its know-how on microbial fermentation and expertise on the use of microbial to create a new business opportunities in Brazilian agricultural sectors. And for us, it can we can do this by close partnership establishment for sales and distribution, but also possibly through joint technology research and development. And thank you for your attention.
Thank you, Jonathan. We have some room for some question from the audience. Is there any question from the participant, please? No question for the moment. I will maybe make the first one to start with. Do you have already some product registered in Brazil? Actually,
not for the moment. No.
Okay, that means everything still need to be done, let's say, on the Brazilian market, and then all the field is open for the partner to come to develop something with Biovitis on the local market.
Yes, everything is open. For the moment, we don't know if it's, or we have an idea, but we can discuss about the best strategy. Is it to introduce our strain or to develop with our know-how strain from a Brazilian soil, strain from agricultural soil in Brazil?
Okay.
So it's open.
You will provide the expertise in terms of formulation and the development of the strain, obviously scale up and formulation as well.
Yes.
Okay. Any additional question? Anything to interact? Maybe nobody. It was clear. Well, Jonathan, you are probably as well available tomorrow for B2B, in case of people is willing to interact with you.
Yes, it will be my colleague Guillaume, but yes, we are available tomorrow. Yes.
Okay, perfect. Then thank you very much. Maybe you can stop sharing your screen. having accepted the invitation. You from Brazil, Sao Paulo, as far as I understood a little bit from our exchange last week. You are from you are the founder of a startup, IAR-do, and you're welcome to present your activity, what you do, and hopefully to find some partnerships with your colleagues present here today.
Okay, bonjour. Je m'appelle Ronaldo. just kidding. I'm not gonna try any French now because it's just not the time to do that. But thank you everyone for the organizers for inviting me and everyone who is taking part of this very nice webinar. It's just funny because I have a hate and love relationship with France. As a kid, especially, Alan Prost was my enemy. I really love it. Ayrton Senna, and then we have Zinedine Zidane in the World Cup, and my God, the French guys are just. But then I did my PhD in Germany, and I used to go to France for the weekend, and I just love it, the culture and the food and the wine, and that's why I have this hate and love relationship. Okay, but just kidding. Let's start with the business. So my name is Ronaldo Dalio. I am the CEO and founder of IAR-do. So basically, just to put in simple words, we are a provider of proprietary biological based products to Brazilian agriculture markets, and we are looking for partners to expand to global markets. So our business model is just that we don't sell the final product. We are not a factory. We are just the very beginning of the chain in which we take an idea, a nice microorganism or a product, and we develop that, and then we transfer this technology to other companies that have a distribution center, that had some market and had some factory to produce the product. So we are really at the beginning of the chain, and that's what we do best. So IAR-do is a spin-off from the laboratory of physiology and biochemical in biochemistry, really related to plant pathogen interactions, from the phytopathology department of University of São Paulo. We are in Piracicaba and not in São Paulo. We are at São Paulo State, but we are in the Piracicaba city. And here we have a very nice ecosystem together with ESALC, which is the School of Agriculture. It was named one of the top 5 agronomy schools in the world. And we are very happy to be here. And there is, we are building a very nice ecosystem, somehow related to the AgroParisTech in Paris. So we have a brand new lab in the technological park of Piracicaba, and it all started in the university, and from there we just mixed expertise. So a little bit of my expertise and the other founder, which is Professor Sergio Pascolati. He has more than 40 years studying, 40 years experience and studying alternative control of plant diseases, and especially regarding to resistance induction through microorganisms and biological control. So together with his expertise and my expertise, which I developed during my PhD and some postdocs, in which we developed a bioinformatics pipeline in which you take the microorganism, you sequence the genome, and then you predict the candidate effector. So you predict some molecules that can manipulate plant physiology. So we combine all that to create IAR-do. So let me talk about the interaction model. So as I told you, we are a company that focus on transfer technology So we develop innovation and then we transfer that to the others. And basically we work with projects. So we have a maximum capacity, a maximum number of projects that we can carry on. And in between this maximum capacity, we divided that 1/3 for development of internal projects. What I mean by internal projects is just an idea or a microorganism that we have, or a nice technology that we have that we want to explore and go from the idea to the prototype. And then when it's done, then it is the time to go to the market and to transfer and to register a product with some partner that can produce and distribute that. So that's 1/3 of our capacity is for developing this internal projects. And the other 2/3 are divided in 3 other business models. One of them is to customize products. It's very common to receive some clients here just saying that, okay, I have a problem with a pathogen and I would like to have a product based on microorganisms that would act as a biological control or as a resistance induction, but this is my problem, I need a solution. And then we go there and we do some customized prototype, and then we transfer this technology for the company. So this is one of the, most of our projects are on this business model, so the customized prototype and solutions. The other business model that we have is just unraveling action mode of bioproducts. So, so Brazil has now a very a brand new law for bioproducts, and a lot of companies are launching products based on plants, extracts, on hydrolysates, in microorganisms and fungi biomass and whatever. So a lot of biostimulants, bioproducts, microorganisms, mix of microorganisms, and this is just very new. And sometimes the companies know that the product is good for some end, for some problem, but sometimes they observe some accessory effects and they just don't know how does it happen. So they come to us, and then we take all of our technologies, we sequence the genomes, we do a lot of bioassays, everything to the molecular level, and then we do the unraveling mode action of this product. So then we can, the company can then do the positioning, a better positioning of this product in the market, or even sell it for a brand new market regarding this accessory effect. So this is another one. And a third one, which is very new for us, we just started a month ago, which is for this new Brazilian law for bioinsumes, for bioproducts. If you want to register a biofertilizer or a biostimulant, you have to do a lot of bioassays. So our government has decreed a law that say all the protocol that you must to perform to prove that your product can be sold in this class, which is biofertilizers. And then we just get the permit from our ministry to perform all those assays and to emit a document, a dossier saying that this product can be registered as a biofertilizer. So this is all the interaction models of IDELAB. So of course, all these external projects, which is customizing products, unraveling mode of action, and issuance of biofertilizer dossier. This is just because we have all the structure to do that, and we have to have some time and some resources to focus on our internal projects, because this is the ones that we are most interested in. So the development of internal projects are really nice because we can have some very good ideas, something that our technology is showing us that it has a lot of success, and then we perceive that and we go on and then we transfer technology. Let me talk about our products. So we have 4 generations of products. We actually have generation 5 and 6, but I believe it's too much to tell right now. Maybe we can if someone is interested, I can explain what are the generation 5 and 6, but it's very easy. On the generation 1, we work in the development of products which are based in only one species of microorganism, which is, so the final product, the prototype will be some spores or cells of a single microorganism. And it's not because it is generation 1 that is it's not good, it's not a nice product. It's just because we have some other generations, because on the generation 1, a lot of prototypes that we have solve some important and some special problems in agriculture. So it's very common to have clients asking for the generation 1 product. On the generation 2, which is one of the most common asked type of prototypes to be developed at IDELAB, and on this generation 2, we just mix some species of microorganisms, and each of these microorganisms has a function such as growth promotion, resistance induction, or solubilizing some nutrient and whatever. It's also very nice on this generation too, because related with BioViti's work, we also combine some microorganisms to develop a prototype which will be like a probiotic for agriculture. So microorganisms that grow really fast and take all the nutrients and occupy the niche in the plant, and then that it can protect the plants from the real pathogens. This is very common. And then on generation 3, we have microorganisms and focused on their molecules. So we have one single microorganism or the mixture of microorganisms, but they are all in a bioreactor. And the final product is not only the cells or the spores of the microorganisms, but it will be the cells or spores together with the metabolites that are produced in the bioreactors. And we are also doing some liquid fermentation that we try to drive the fermentation in order to produce some more of a metabolite or a protein, for example, and so that we can have a product which is complete, which would have the cells or spores, the microorganisms alive, but we have also the metabolite or the protein that can have a function through the plant physiology or through protection to other plant pathogens, for example. So this generation 3 is a very nice technology and a very nice type of prototypes that we are developing here. And the 4th generation, which is the one that I am most focused in, is when we perceive that some microorganisms are secreting a molecule or a protein or something, and this molecule has an effect. And then we can have the genome of this microorganism sequenced, and through a bioinformatics pipeline, we can identify this molecule, and then we clone this gene in a bacteria or in a yeast. We put them in a bioreactor to produce this active ingredient. And then the final product will be not microorganisms anymore. There will be no cells, no spores, but only the protein or the metabolite, the active ingredient that can be sprayed in the plants, that can be treated seeds, and and can be used globally. It's not just only for Brazilian, because Brazilian markets, because you have to have the microorganism registered and the interaction with plant and host. So this generation 4 of prototypes are interesting because you can use it everywhere. You don't need to have it a very special logistics for having a product which is in a specific temperature. You don't need to have interaction with host and no host, because it's a protein, it's a metabolite that you can easily apply by foliarly or treating seeds. So it's a very nice type of prototypes that we develop here. So talking about our microorganism library, we have access to more than 800 species of beneficial fungi and bacteria. So this is this library was from microorganisms that were isolated from tropical and semi-arid areas in Brazil. So they were mostly isolated from Professor Pascolati during his career, and also because a decade ago or something, Professor Pascolati coordinated a huge project in Brazil called Cisbiota, in which we have we isolated beneficial microorganisms for several biomes, and then we tested the ability of these microorganisms to induce resistance, to promote growth and productivity, and also for biological control. So between this library, we have a very nice list of microorganisms that are already studied and have already some effects. Most of them are not registered, are not being used at the moment, but we have access to this, and we can perform some prototypes depending on the client, or even through our internal projects development. So what are our technologies? So every time we have a new microorganism, we apply systems biology to that. So we first start with sequencing the genome of course, the microorganism has to have some appealing, something that took our attention, some effect in vitro or even in plant. But after that, we go to genomics and bioinformatics. So we sequence the genome, we apply our bioinformatics pipeline, and we identify the secreted candidates, the effector molecules, the molecules that can act upon plants and upon to the microbiome. And after that, we perform transcriptomics, proteomics, and metabolomics to assess the role of these metabolites and how this microorganism acts. And through the end, we go to the phenotyping platform. So we just closed a partnership with another company from Czech Republic, and we have a platform for phenotyping from high throughput screening of biostimulants, of compounds, metabolites from microorganisms, and bio-based products. So for one round from this platform, we can test more than 2,700 compounds, for example. So it's very fast, very easy to do. Use some other plants. I can talk more about the phenotyping platform, the high throughput screening phenotype, if someone is interested. So these are our technologies. I am happy to announce here that we just closed a partnership with Capsera, which is in this webinar. Antoine's here. I'm just very happy to announce that we are sharing our technology. So you use our microorganisms, our bio-based products, together with the encapsulation technologies of Capsera. So it's already happening. So the integration of Brazilian and French companies, we are very happy to announce that. And just to go through the end, what are the main focus focused areas that we are working, that we are developing products. So mainly biological control, induction of disease resistance. This comes from the expertise of Professor Pascolati. So this is one of the pillars of IDEA Lab. So we do a lot of prototypes related to enhance the plant defense system. So it's not really biological control, but you apply it, the microorganism or the generation 3 or 4 products to the plant to enhance its natural defense. We have also a lot of prototypes that are related to growth promotion and plant physiology, and also some microorganisms that can be really useful for to apply to induce tolerance to abiotic stresses such as salinity and drought stress. So we have a very nice mix of microorganisms that can be used to readapt plants after those stresses. So these are the main 4 pillars in which we focus to develop our product, but we have some others such as probiotics, and also very similarly to BioVitis, thinking account for polluted areas or wasteland areas and whatever. So these are some of our partners. So we are just beginning our so for you to have so this new lab that we are right now, we just was opening 2 months ago. So we are working in the past years, but this new lab is very new. So but we already have some very nice partners. So our lab, which is this new one that I'm talking about, is 150 square meters, and we have also 2 greenhouses with 100 square meters, and we can use those greenhouses for developing those products, for doing that bioassays. And we have partners throughout Brazil, mainly São Paulo state, to do the field trials. Our team is a very small team. We are in 13 people right now, but we have 5 PhDs, biologists, bioprocess and biotechnology engineers, and agronomists. So we are a very small team, but very efficient. And we are looking for partners to expand our capacity to develop projects, and also to expand our infrastructure. I was really happy to show a little bit about IDEA Lab. Feel free to contact us, to send a message. So we are on Instagram. We are also on LinkedIn. If someone is interested, please feel free to contact myself. And also I will be available for questions right now and for tomorrow, the B2B meetings. So if anyone is interested, please just say hello, just get in contact. Thank you very much.
Thank you, Ronaldo. Thank you for this very nice presentation. Is there any question from the audience? One of you that is willing to discuss a little bit and to have more precise, let's say, idea about the work of Ronaldo? Nobody. Just to make sure, then I will start with one question. You are a service provider, but what is your business model to stay as a service provider, or you have the intention in future to register and to distribute biocontrol product as well, or to sell just biocontrol product in Brazil or in South America in general?
Great question. We are, so as I told you, we are just beginning with this new phase, with this new lab. And right now we are just focused on transferring proprietary products. So we are transferring intellectual property to our partners to produce and to distribute that. But we are open to partnerships. In the future, I really can see that if we have some really nice products, maybe it's a better idea to just keep it to ourselves, and to open some factory and enter the marketing as our own brand, as our own distribution channels. So we are open to that. Right now, it's not the model, because since we are not, we don't have any facility to produce or any channel to distribute that. We are not engaging on that right now, but we have some nice prototypes that would fit to that. So we are open to partnerships, and we are open to further discuss. And also, it was really a challenge to start a new lab and start all those new projects. It's growing really fast. And right now, if you want to open a factory or a distribution channel, it has to be with a partner, because we have no time to enter in this new challenge. So maybe if someone is interested, please contact me.
Okay, thank you. Thank you very much.
So hello everybody. So Brazilian partners are in the morning and French in the afternoon, so it's quite nice. And I'd like to thank as well very much for the invitation to present today in the session of Biocontrol. So I'm Hélène Lacroix. I'm Associate Director of the Fédération Condorcet, and I'm going to present today so first of all, a bit of the federation, and then I'm going to focus on biocontrol and present 2 examples of projects that we had within the federation. So here we're based in the north of France, so basically in 2 regions of the north, and as well in Wallonia, a region of Belgium. And our expertise starts from sustainable plant production, plant biomass transformation and processes, innovative agro-sources products and materials, and societal conditions for sustainable bioeconomy. So we're really from the production of processing and processing of the whole plant. So our objectives are create synergies between academic research members, pool resources and equipment, support implementation of new educational projects, and integrate new members and become an international reference. So more in detail about the federation. So we are 30 labs, 14 technical platforms, and about 700 researchers. So as I said, so we're based in 3 European regions, and we're funded by 3 universities and labeled by CNRS. So I don't know if you see my narrow here, but we have the University of Lille, we have the University of Liège, we have as well 3 engineer school, AgroParisTech, Centrale Supélec, and Unilassal. We have the University of Reims here, the University of Compiègne, and the one of Amiens here, and the one of Littoral Côte d'Opale, so in the north, Calais and Dunkirk. And recently we had as well a Brazilian partner that joined us in October 2018. So it's the group Sinai Cetic that is really focused in chemistry and industrial biotechnology. And I saw today, so it's maybe presenting in another session. So our expertise, I'm not going to go into all details, but I have highlighted actually the expertise that we have in biocontrol. And so I'm just going to go through the main expertise. So plant biology and biochemistry, microbiology, environmental sciences, soil sciences, agrobiochemistry, agromaterials, electrochemistry, process development, and societal and economic aspect of bioeconomy. So our platforms, we have platforms in analytical, so in chemistry, in catalysis, a calculator, quite important at the University of Reims, a platform in nanotechnology, as well in microscopy. So we have 2 platforms on that. We have as well on photosynthesis, so for the measurement of carbon metabolism, but as well for the gas exchanges. We have platform in environment, and as well one in flux cytometry. We have a big center at the University of Amiens on biology, on molecular biology, sorry. And of course, we got in almost all the partners, they have greenhouses, and the University of Liège, for instance, have phytotrons as well. So what does a research federation? So we have 3 actions. One is the animation. So of course, we have 700 researchers in the federation. So we need to gather them together and organize events and working groups and stuff like that to actually exchange. We provide as well visibility of the community. We create communication tools, databases, website, newsletter, and we do as well disseminate information within the federation, so about calls, jobs, events, whatever. So we have as well an action in supporting project ideas. So every year we have a small call that's actually, well, for funding a 1-year project idea between 2 partners, labs or platforms. And this helps to actually build bigger projects, so on the nationally or internationally. We do as well help building European projects. So for that, we identify calls for the federation. We bring greater visibility at EU events like Info Days and brokerage events in Brussels. And we as well help building consortia. We are as well in relation with the French governments to contribute to EU work programs. So for instance, Horizon Europe. So now focusing more on biocontrol. So the labs that are involved in that field well, we have 93 researchers, 7 laboratories, and 10 technical platforms. So here I show you all the same map with all the different partners in biocontrol. So they're mainly in the field of plant pathology. So looking at really understanding the relationship between plants and microorganisms, beneficial and pathogenic. Apart from the lab AgroParisTech, which is more focused on, well, it's more a chemistry lab, so it's more interested in the aspect of the degradation of lignocellulose, and for that, to get maybe new added biosource products from that could be applied in the biocontrol field. So then I'm going to focus more on 2 different projects that have been, well, they've been started like 4 years ago and they're finishing now. So one of them is ELISA project. So the title is Optimization of Rhamnolipid Mixtures with Antifungal and Eliciting Properties for the Protection of Rapeseed. So we're really working on a molecule that's called rhamnolipids that's produced by Pseudomonas aeruginosa, and this compounds has several properties like it is biodegradable, non-toxic, biosurfactant, and has antifungal properties and stimulate plant defense activities. So in that project, they focus on fungal attacks on rape seeds, so from Sclerotinia sclerotirum and Leptophoria maculans, which is the fauna. The objective of the project is really to reduce actually the conventional pesticides that are used. So I put you the contact, the coordinator contact, donc Sonia Ripa. This project has been funded of about 1.5 million euros and for 36 months. And it has the particularity as well to, in the consortiums, to bring together industries and as well producers and researchers. So here I label with the little leaf the partners that are in the federation. So it's the Induced Resistance and Bioprotection Plant Lab, so RIBP at the University of France, and it's the Enzyme and Cell Engineering Lab at University of Amiens and University of Compiègne. So the main objective of the project was to actually get different type of rhamnolipid mixtures. So with the first characterization, and then it has been tested for antifungal and elicitor activities on the microtite plates or on the agar plates. And the best results were transferred to plant protective assays, still in controlled conditions, so in the lab. And then from then on, the best results of that went into field trials. So you can see as well, I put the reference of the publication that came out in 2020 in phytopathology. So you can go to that publication if you're interested in. So the second project I'm going to talk about is Smart Biocontrol. So it's a project that's been integrated, been financed by the Interreg program. The consortium is quite large because it's 26 partners. The funding was 20 million euros and it was for 36 months. So here again, so it's a huge consortium that brings together researchers, industries, and producers. And so that was the quite interesting part of the project, is that really to bring new solutions from lab to field. So I showed as well the partners of the federation that are involved in the consortium. So more in details about Smart Biocontrol. So it's a big portfolio. It's not just only one project, but it's 5 projects in the same time. So that's why the budget was so important. I focus on BioScreen project because it's the one that actually was the objective of the project was to develop a high throughput platform of new biocontrol products. And then you had BioProtect. That was more to assess the new biocontrol products, the effectiveness of those products in agronomic conditions. Then BioProd was more to develop production of those new biocontrol products on industrial scale. And then Biosense was to ensure monitoring the field, like to be able to detect pathogens, but as well the biocontrol agents. And the last project was really the coordination project. So BioScreen was based on two families of molecules. So one, it was the lipopeptides from Bacillus subtilis, and the other family was rhamnolipids from Pseudomonas aeruginosa. Well, in the lab, those molecules have already proven that actually they were effective against several diseases for lettuce, leek, tomato, potatoes, and wheat and grapevine. So it started from that, and that's why they really wanted to implement those products on the field. So to remember, in conclusion, so we're a seniors federation of 30 labs, 14 platforms, 700 scientists in the field of bioeconomy, so larger than the Biocontrol only, in the north of France and Wallonia in Belgium. We have expertise and skill in exact natural and social sciences. We have technical and technological supports. We have a strong relationship with industry, with the IAR cluster, and as well the European Bio-Based Industry Consortium. And we aim to develop national and international projects. So 1/3 of Condorcet's research activities are in the field of biocontrol, and the biocontrol expertises are plant pathology, agroecology, chemistry, epidemiology, soil, and plant physiology studies. So thank you very much for your attention.
Thank you, Hélène. Thank you. Thank you very much. Do we have any question from the audience, maybe? No real questions? Okay, that was pretty clear, I presume. Yeah. Hélène, can you tell me a little bit more about, let's say, the arrival of Senai, let's say, in your the Brazilian partner in the federation? What is their role and what are they bringing to the federation?
Well, the thing is that it's been a relation. Well, an interaction that's been done by Christophe Clément. I remember maybe when you organized for the first time like this, a gather with partners from Brazil, and they have interactions with different laboratories in chemistry. So with AgroParisTech, and I think as well with the IMR at the University of Compiègne. Okay.
And what are you looking further, let's say, as cooperation in Brazil, actually, for the federation? What could be of interest for your federation now?
Well, a lot, because I mean, there's a lot of things to do together, I think. Well, we need to develop more and more international partnership, and as I said, I mean, we have 1/3 of our activities is based on biocontrol, and from the consortium that's been done with Smart Biocontrol, I mean, it should be logic to actually go even further and spread out to other countries that could be interested to implement this kind of technology in their agriculture.
Okay, thank you. Thank you very much.
Thank you, John, for organizing this webinar and giving me the opportunity to present Capcera. So I'm Antoine, CEO of Capcera and founder of the company, and I will introduce our company to all of you. First, the context in which we created Capcera, you probably all of you know this. We are facing an unprecedented challenge according to FAO. We will be 10 million people on Earth in 2050, and we have to improve our production capacity from 70%, and we have to do it in a sustainable way. And a major challenge is to switch from an agriculture system based on chemical inputs to an agriculture system based on biological input. It means biosourced and biodegradable. So it's a great switch and a great opportunity for a young company bringing innovation on the market to create new business, and this is what Capcera wants to do. We are operating on the market of inputs, and you know it, fertilizers market and pesticides market together worth 200 billion euro, and only a very small portion of this is dedicated to bio inputs, I mean biofertilizers and biopesticides. Why is it so old? Despite everyone, politics, consumers, some key players in the field wants to promote a more sustainable model. it's mostly due to the poor efficiency of bio-inputs in field conditions. Most of these products on the market are dedicated to greenhouse production, where you control the condition of production. Once you go into the field, there is an hyper-variability of condition of application, various soil composition, climatic conditions, and it's much more difficult to have performant product. It is a matter of formulation. So this is exactly what we are addressing at Capcera, bringing innovation on formulation of bioinputs to enable them in field condition. And our aim is to become industrial. We do not license our technology. We develop products through partnerships with academics or companies that bring the active ingredient. It can be microbes or natural extract or pheromone. And we encapsulate them with innovative capsules to create a performant product. And at the end of the day, we will produce industrially, at least we will formulate this product industrially. Today, we are a little bit more than 15 people. We are based in Paris with 140 square meter lab and office. And the team is a combination of skills in expertise in microfluidic, microbiology, and agronomy, everything we need to bring our technology on the market. Our ambition is to create new standard of formulation, specially developed for biofertilizers and biopesticides. It's not a well-known technology developed for agrochemicals or for food industry. It's new technology specifically developed to protect natural biodegradable ingredient for field application. And the idea is to make sure our technology will match all expected features for the market: stability over storage at room temperature, easy usability for farmers, compatibility with current equipment on farm, and of course performance in field. And if we match this, we will create value in the product and share it with the value chain, marketers, retailers, and farmers. We are bringing a new technology of formulation. It's a microfluidic encapsulation process. We are, let's say, upgrading something well known from microbiologists, the alginate technology. You probably know this. It's very easy to encapsulate alginate microbes in beads based on alginate. You mix a liquid solution of alginate, it's sodium alginate, with a tiny amount of bacteria and some sugar and amino acid to feed the bacteria. And then you drip this solution in a calcium bath, and each droplet going into the calcium bath is converted into a nitrogel of alginate, trapping a few amounts of microbes and sugar and amino acid. So it's a field sphere, a beads, that is quite performant for some application, but not enough performant for agriculture purpose in field. First, it's not enough concentrated in bacteria. And second, it's extremely difficult to control the release of this bacteria in the field. You have to dissolve the bead, and it's extremely difficult to do it in field condition. We develop a new kind of way to use this technology by using micro-injector that are able to combine two liquid solutions. One is an highly concentrated solution of active ingredient, and the second one is still alginate solution. And we create droplets with an inner part composed by the active ingredient solution and a thin shell of liquid alginate in the surface of this droplet. And we collect them into calcium baths to trick the gelling of the shell. And at the end, we get not a beads, but a balloon with a liquid core. And the liquid core is exactly your active ingredient solution. And the shell is a thin shell of alginate. And we can produce like these capsules containing high concentration of microbes in liquid media or pheromone in oily solution. We can encapsulate anything that is liquid and adapt it in size to our micro-injector. Of course, this technology is patented, and we have exclusive license on the 4 patent families worldwide for agriculture purpose. Alginate is a perfect material for sustainable agriculture. It's a reliable supply chain. It's biosourced, biodegradable, already approved on many markets, especially in Europe and probably in Brazil. And we can produce capsules like you can see on the pictures with a thin shell of alginate in the range of 10 to 50 microns. and the size of the capsule can produce a capsule in the range of 100 to 500 microns, even bigger, but for agriculture it's better to be 100 and 500 microns diameter, with high concentration of bacteria like on the picture. And with this technology, with like universal, we can encapsulate anything that is liquid. We can also customize. It means adapt the recipe, the composition of the core of the shell or around the capsules to optimize the product, to specify the product for the active ingredient and the purpose at the end. And in all our partnerships today, we are always addressing one of these topics: protecting the active ingredient from various stress during storage or UV exposure, allowing combination of this natural active ingredient with a conventional product. It's important topics for the transition between chemicals and biological product. Today you cannot mix together live microbes with synthetic pesticide. Very often you will kill the microbes. So we are developing capsules to make this combination possible. And finally, to control the release of the active ingredient in the field at the application or after a few days or a few weeks. Since the beginning, we have a great start. We incorporated in January 2018. The first year we secured 2 partnerships with industrial and some funding through a loan to entrepreneur. In 2019, we raised some money from French VC with a seed round, and we get 700 euros, and we win some prizes in France. The more important one is from InVivo, who is the biggest cooperative of farmers in France. And more recently, we get a grant from BPI France, 1 million euro grant to support our scale-up project and development of product dedicated to grape wine. The company now is well structured with a board, including Jérôme, the scientist that develops the technology, and I. We are the 2 main co-founders of the company, and independent board member that advise us to reach the market efficiently with performance products. So Cédric, coming from the fertilizers market, and Yvonique from the pesticides market. Cyril is representing the investor in our board. Now we are 16 people exactly, and let's say half of the team is dedicated to bringing our technology to industrial scale. So the first objective is to scale up the process of encapsulation. And the second is to develop new capsules from this simple concept of alginate microcapsule. We are developing different kind of capsules with a different purpose, addressing market needs of the sustainable agriculture. And the second part of the team is dedicated to product development using those capsules in partnerships with academic, lab or startup or big company, bringing their active ingredient and trying to make with us the best possible product. For more than 2 years now, we are accelerating on technology development, especially on building our portfolio of innovative capsule. Still the same slide, summarizing what we are doing with different specific projects on each of the 3 categories. I can highlight 2 important ones. The first one, UV protection, is something really recurrent with alive microbes for bacteria coming from rhizosphere or some virus to fight against against disease. You need to protect those live microbes from UV. So we are developing capsule for this. And we are also developing capsule for slow release of fertilizer, especially for organic fertilizers, and second one for pheromone diffusion to develop sprayable formulation. It's 2 important aspects of the sustainable agriculture. So if you're looking for a solution for this, please, you're welcome to discuss with us tomorrow at the B2B meeting. We are also developing innovative products. So a quick overview of what we are doing. So as you can see, we can develop product for any crops. There is no limitation, and any kind of product, biostimulants or biopesticide. Today we are mostly active on biopesticide for cereal and grape wine due to our location in Europe and especially in France. But we have also some partner with Brazilian company, as you know now, and North American company for other application. Concerning our scale-up plan, we have made huge progress since for the last 2 years. From the first, let's say, stage of the technology when we incorporate the company, we multiply our production capacity by 80 times. With our current lab pilot, we produce enough capsules in a day to perform several trials over hectares of fields. And we have a clear plan to go to the next step, which is a pilot plant. Thanks to the funding of BPI France, we will implement this pilot plant next year in France. And we have also already a clear plan to reach higher capacity within the next 5 years. What we know from our capsule now, first, they are fully sustainable. We only work with ingredients. The most important one is alginate, but other ingredients used in our formulation are biosourced and biodegradable. We know that we can encapsulate any kind of active ingredients for agriculture purpose. It can be microbes, virus, fungi, bacteria, pheromones in oily or aqueous phase. We can also encapsulate emulsions. We always reach high concentration of active ingredients, especially for microbes. It's roughly exactly the same concentration as the wettable powder that you have on market now. They are easy and safe to use. The active ingredient is inside the capsule, so there is no direct exposure of the farmer to the product. And even if we dry the product, it's a very safe powder because there is no small dust, so it's very safe to handle and to resuspend into the sprayer. And the resuspendability of our dry capsule is very easy. And finally, we can apply those products on seed, on soil, and through foliar treatment. It's compatible with any kind of treatment. We have some promising results that I can share with you in laboratory concerning stability improvement over storage, compatibility with chemical pesticides of bacteria, or pheromone diffusion in lab condition. And also in field and greenhouse, we performed last year a lot of trials in Europe, especially in France, in field and in greenhouse on various crops. And we validated that it's compatible with sprayer and spreader on the market. We have now a good view on which nozzle you have to use to apply our capsules. We know that there is no secondary effect of our crops. They are perfectly selective, and some good results on cereal for seed treatment and biostimulation. What about Brazil? I'm looking for a partner in Brazil, and you know it, we found one. In fact, I went to Brazil 1 year ago, thanks to Business France and Claire Meunier, who introduced Business France at the beginning of the webinar. And I went there exactly with this picture saying I'm looking for a great partner to develop successful partnership between Brazilian guy and French company, like Ronaldinho and Rai with Paris Saint-Germain. And I found my Ronaldinho, it's a Ronaldo from IAR-do, and Rai, it's Sergio. And we recently signed our collaboration agreement after 6 months of discussion. And we agreed to combine our skill and expertise to develop a great product for Brazilian market. So if there is any people interested in working with Capsera to develop a product for the Brazilian market, you are more than welcome. And if you want to support our partnership with IAR-do, it's possible to discuss this with Ronaldo and I. So feel free to contact me. I will be available tomorrow for the B2B meeting. So feel free to ask any question now or to set up a meeting with me tomorrow. Thanks very much.
Thank you, Trent. Thank you for this nice presentation. Very clear to me, but I knew already your technology and your company. Then it's very easy for me. We have time for questions. Is there any question from the audience, please?
Yes, from Biovitis, Jonathan Gerbord. Thank you for this very interesting presentation and the promising technology. Do you have experience on combining microorganisms in the same formulation?
We have, yes, we have experience. We did some trials with two different strategies. The first one, it's very easy. It's to develop single formulation. I mean, 1 microbes per capsules, and you produce different formulation of capsules, and then you mix the capsules together in different ratio. And it could be a way to adapt the ratio to the some geographic areas. And the second things we tried, but it's a case-by-case, let's say, scenario. It's to encapsulate together the microbes in capsules and sometimes to grow them in capsules. But you need to, let's say, to adapt the condition of growth. And it's possible to achieve interesting results like this. But like I said, it's a case-by-case scenario.
Thank you.
Okay, thank you. First of all, I would like to thank you for the opportunity to be here and to share what we are doing on biocontrol at Embrapa. Just to know who don't know Embrapa, Embrapa is a Brazilian agricultural research corporation. We have a little bit more than 40 units all around Brazil working with agricultural research in different areas. So we have some regional centers, national product or commodity centers, and the national thematic centers, like where I work at Embrapa Environment, that it's a unit that work with the environment issues related to the agriculture. And so we do at Embrapa Environment transdisciplinary research based on impact assessment and the environment management of agriculture. So here we work with different mechanisms of impact assessment, life cycle assessments, carbon footprint, and ecosystem management. We work with development of agro-environmental quality and sustainable production systems. And we deal with global climate change and agriculture, all the relationships about this, and the bioprospection, and environmental biotechnology, where biological control is inside. But all of these 4 areas are connected in our work. So to talk a little bit about what we are doing on bioproducts, on biocontrol, we work in the way to bring innovation on biocontrol, and we work in this sense of the convergence of knowledge about not only the biological issues, but all the new technologies that are coming, and this connection of these bits, genes, neurons, and atoms, or all the new science can be allow us to understand the interaction between plants and microorganisms insects and the other elements in the nature. So this connection points to the new paradigm on biocontrol, like the new biocontrol agents that we are selecting in our bioprospection. We are working not only in the sense of one-to-one microorganisms against pathogen, but the microbiome system too. I'll show a little bit about that. Advanced fermentation process. We just have a new patent on optimization process of submerged liquid fermentation for fungi. That it is very good results in scale up of the fermentation of fungi propagals. We are working on new modes of action, not only biocontrol, but stress resistance, induction resistance on plants, growth promotion, and so on new formulations and on application technology too. And here is just an example of application technology that we are working for biocontrol. It's electrostatic pulverization that allow a very good coverage of fungi propags on the field that give really a very nice control and allow us to achieve the biological of. So we believe that biological control is really a very nice technology that has a great future on phytosanitary systems, on plant pests and disease control. So it's no more the ugly duckling of the plant health, but it's now really a very robust and scientific technology. So we will work to develop this. Just to show a little bit what is going on in Brazil about biocontrol. This is the evolution of the registration of biological control products in Brazil. We have a little bit more than 300 bioproducts now registered in Brazil. And about 65% of that it's microorganisms, and 20% macroorganisms and semiochemicals and biochemicals to complete this list. But it's really a very growth registration process. And this is what we have, the main organisms that we have registered in Brazil, including virus, fungi, mites, nematodes, bacteria, and insects, parasitoids, and so on. So this is what we have registered in Brazil nowadays. But this market is growing very fast. So we are facing 15% increase in the last years of the market of the biological control in Brazil. So what are we doing in biocontrol at Embrapa? So Embrapa is a research and development company. So we do not sell products, but we developed technology and products and share these with companies that will be the proprietor of the product that will register it and sell the product. So we do biodiversity prospection. We do strategy for selection and the trials of the performance of the biocontrol agents. We develop strategies for integrations of the biocontrol agents is integrate pest management. We study the environmental and socioeconomic impacts of the biocontrol agents. and we work with the strategies for adoption and to technology transfers for the industry and for the farmers. So we are working on biocontrol of pests and disease, inoculants of our growth promotion, and some inoculants for abiotic stress like drought tolerance, salinity tolerance, and so on. On biodiverse prospection, we have a collection of microorganisms with about 20,000 access, microorganism access from different biomes in Brazil, since Amazon, Caatinga, Cerrados, and so on, and some other environments that is we call environment stressed environments, like we have microorganisms from high salinity areas, like some soils we have from Antarctica, and so on. So we have a really a huge collection. About 10 to 15% of these 20,000 microorganisms are already characterized and analyzed. So we are using this collection for development of new products for biological control and for other industrial and agricultural use. So this is just some view of that. And we work in develop called this typical biocontrol products, one-to-one approach based on a very no process that was shown before here by different people before me. But we work to on other approach, like the microbiome approach, the interaction of these organisms on the field, and understanding not only the organism at sea, but to understand the connection of these different functions, different genomic functions that are associated with different plants. So we do working aiming to understand the impact on the physiology, metabolism, nutrient acquisition, production of different metabolites, and plant growth hormones, for example. And we try to understand to the some challenges for the biological control industries in Brazil. Brazil has some peculiarity that grinds the dissemination of biological products. In addition to the high number of pests for the tropical climate, we have very large areas. We have 2 or 3 yields by year, so we don't have a winter, a real winter in Brazil. So we have this green bridge all over the year. So this difference has an impact in the development of the biological control products in Brazil and the distribution of this product. So we are facing these challenges and developing technologies to help companies to make these products feasible for Brazilian markets. So we working on training of professional for development of and use of biological control, like specific requirements of handling and application. We build the public-private partnership for joint development of products. So mainly based on the transfer of knowledge and the superior genetic material strains of microorganisms for companies that are able to develop products and to register products to the market. And we have a very confidence and prestige as a science-based innovation company. So we promote biological control using this science-based innovation process. So just to bring some final remarks here, biocontrol is a big opportunity, not only in Brazil, but the Brazilian market is really big and growing. So we believe that this technological convergence is a trend in all science. It is include biological control. So biological control must go to the agriculture 4.0, the digital agriculture. Biological control must be inserted on that. It's really a very nice time because the consumer, the public is claiming for more sustainable food production. So biological control comes to fill a gap on that. We have here a thriving innovation existing really developing in Brazil with the emergence of innovative companies that we can support, that we can give support as a science company. And so Embrapa's biocontrol portfolio brings partnership opportunities at all stages of new product development, since the bioprospection of new strains until fermentation and development process and until the market of these products. So this is my contact here. I would like to again to thank you for the opportunity to show a little bit about what are you doing at Embrapa for biocontrol. Thank you.
Thank you, Marcelo. Thank you very much for this nice and very complete presentation. Is there any question for Marcelo? No question?
Yeah.
Everything was clear?
Yes, Joan. Antoine from Capsera. Thanks, Marcelo, for your presentation. It's still difficult for a company like us to understand if it's easy or very difficult to register the product in Brazil. So I heard very different story depending on who is talking. So could you maybe share your knowledge or experience in this field with us?
Okay. It's really not a trivial to have registration of product Brazil, but in the last years that was really a great advance on our legislation and on how to register the product in Brazil. And now this year was launched a bioinputs program by the federal government that will facilitate, that will make easier the registration of products. But we have some problems like that with exogenous microorganisms, like that you cannot bring microorganisms from outside of Brazil without risk assessment. So if you register a strain that was isolated from Brazil, it's easier than to bring external product because they need to have some risk assessment studies. But now we have some companies in Brazil that give support for registration process. So if you are interested, Anthony, we have partnership with one of these companies that give support for registration process, that conduct all the documentations and all the trial that we need to make a registration in Brazil.
Okay, thank you very much, Marcelo.
Thank you, Marcelo.
You're welcome.
Any question? No question? Okay, Marcelo, thank you very much for your presentation. It was very nice. This was the last speaker for this session. Thank you all of you for having participated to this session and to this webinar. Before leaving the webinar, we would like you to join maybe the last session, ongoing session. There is the session still process and technology that is open, and that could be of interest for certain of you, especially because the last one are on biotechnology as well, then it could be of interest. And we would like to invite you to join that session. And from that session, we are going to finish over there, because this will be the last session to finish. Thank you very much in advance. For those who are not going to join that session, hopefully you are going to have a very nice B2B meeting tomorrow. That will be the rise of a new partnerships between Brazilian and French company. And we stay at your disposal in case of need, and in case you are willing to reach and you could not reach one of the other person. And you want, let's say, to reach someone that is not present at the webinar, but we have with Renata on site in Brazil, and with us here in France for the Brazilians who are looking to reach French company, very interesting possibility to enhance and to faster, let's say, cooperation. What else to say? Just to invite people that are interested to network at the international level in Europe next year during our Plant Bioprotect Congress. It will be in November next year, normally in Liège. We are, as I said in the beginning, waiting the final confirmation from the location. And it will be on site. We hope that COVID will be finished by November next year. And a call for paper will probably soon be launched, let's say, by our organization. And Hélène Lacroix from the SFR Condorcet and RIBP of URCA, co-organizer with us of this event. We will be very, very happy and very honored to receive some Brazilian partners, let's say, during this event. Now it's probably time to leave the room, and while you will be on the main room, we will forward you back to the process and technology room. Maybe some of you who has the last version of Zoom might be able to do it directly from their Zoom system. It's just a button that you have at the bottom of your screen. Feel free to do it, and you're welcome to see each other again in the process and technology room. Thank you very much, and have a nice afternoon for those who I will not be able to see anymore. Thank you. Bye-bye.